Assalamu alaikum. As we know, forensic DNA analysis is an extremely powerful investigative technique that has become, in many ways, the standard by which other forensic sciences are measured. Forensic analysis or forensic DNA analysis used to identify individuals using genetic samples. Basically, the system uses a set of identifying numbers to match two different samples. Forensic DNA analysis has played a crucial role in the investigation and resolution of thousands of crimes since the late 1980s. The concept was first designed by Sir Alec Jeffreys at the University of Leicester in 1985, and today it is a standard practice in conducting investigations for crimes such as murder and rape. So my topic is DNA forensic analysis, in which we study about DNA structures and function, forensic and DNA. Collecting DNA samples, determining a DNA profile, how it's done, DNA profile in solving the crime, DNA databanks. We will discuss about some case studies like how to collect a sample in a bomb blast attack or in a sexual assault case. So, while moving on, this is a presentation presented to Dr. Hammad Majeed, presented by me. The first topic is the information about DNA structure and function. DNA is deoxyribonucleic acid that contains genetic information. DNA codes the protein that our body make, which are necessary for survival. Thus, DNA is a code for making proteins. DNA also determines how much of these proteins each cell makes. The order of amino acids determine what type of protein is made. Some common proteins are hemoglobin that carries oxygen from lungs to cell, insulin that regulate metabolism, many types of enzymes that catalyze reactions in the body such as breakdown of sugar for energy. So, when human cells are present in biological evidence, their chromosomes can be examined to determine whether the evidence comes from a male or a female. The analysis of chromosomes is known as karyotyping. DNA fingerprinting, also known as DNA profiling, is used in criminal and legal cases to determine identity or parentage. Trace the inheritance of genetic disorder, identify the origin of a blood, semen, or saliva in a sample, and identify victims of war and large scale disasters such as plane crashes, tsunamis, and hurricanes. 3 billion bases in human DNA, 99% of DNA is identical among individuals. 1% contains significant variation. Each person's DNA profile is unique except identical twins. So this is the structure of DNA. DNA is actually double helix twisted structure. uh, twisted ladder. The DNA ladder is made up of building blocks called nucleotides. Okay, the two strands are held. The two DNA strands are antiparallel. The two strands are held together by hydrogen bonds formed between the complementary bases sugar phosphate backbone side as the sides of ladder, nitrogenous bases rungs of ladder. Next, we will discuss about nucleotides. Nucleotides are biological molecules that form the building blocks of nucleic acid. DNA and RNA, they serve to carry packets of energy within the cell, ATP. Nucleotides play a central role in metabolism. A nucleotide is composed of nucleobase, nitrogenous base, a 5-carbon sugar, either ribose or 2 deoxyribose, or one or more phosphate groups. 4 bases of DNA. The nitrogenous bases found in nucleotides are classified as pyrimidines or purines. Purines have two ring structures. Pyrimidine has one ring. This is the source from where this information is collected. Pairing of pairing of base pairs. Adenine always pair with thymine and guanine always pair up with cytosine in the DNA structure. The next question is that okay, this is the prominent diagram of DNA. The next question is, where is DNA? DNA is present in the nucleus of the cell. DNA in the nucleus is packaged into chromosomes, 23 pairs. DNA can be recovered from any substance that contains cell. All types of cells in our body contain a copy of the same DNA. DNA holds the instructions to make all things in your body work properly. The next is forensics and DNA. Forensics is the application of science in a legal setting. An example of modern forensic evidence is the use of DNA profiling. Sources of DNA include blood, hair, semen, saliva, bone, and tissue. Collecting DNA samples, DNA can be isolated from a wide range of evidence left at a crime scene, from skin, hair, and semen sample to bacteria in dirt. Forensic scientists, for example, those who work at environmental science and research, are required to collect biological material from a crime scene. Blood is an excellent source of DNA. It is collected from the white blood cells, mature red blood cells, and do not contain DNA. DNA can also be obtained from the heads of individual sperm cells, from hair follicles, or from any other cellular tissue. The biological material used to determine a DNA profile include blood, semen, saliva, urine, feces, hair, teeth, bone, tissue, and cell samples that may be used. Investigators collect items that could have been touched or worn by persons involved in a crime. The following items may contain DNA material like masks, hat, gloves, clothing tools, weapons, sexual assault evidence kits, undercloths, bedding, dirty laundry, fingernails, carping, cup bottles, cigarettes, toothpicks, toothbrush, facial tissue, hairbrush, eyeglasses, tape, ligatures like rope, wire, cords, stamps, or envelopes. The best evidence occurs when a person's DNA is found where it is not supposed to be. For example, consider a breaking and entering that occurred in a residential area. Near the point of forced entry, a knit cap was found which the homeowner confirms was not theirs. Survival head hairs were recovered from the inside, one of which had a root with tissue attached, which made it possible to obtain a DNA profile. The DNA profile was used to identify the perpetrators. For example, imagine someone has broken into the school office uh, where exam records are kept. Perhaps some hairs dropped off without a person realizing, or maybe the person cut their arm on the broken window when they entered, leaving a small trace of blood. It would be up to forensics team to find these samples and collect them for the analysis in the lab. This picture elaborates that a crime scene investigator uses a swab to collect blood from a crime scene. A cigarette butt found at a crime scene may contain valuable DNA material in the dried saliva. DNA evidence from both the victim's blood and the perpetrator's skin cells may be available from this hammer. Okay, as the technology advances, forensic scientists are able to analyze smaller and smaller biological samples to develop a DNA profile. For example, if a person touched an object or weapon, skin cells may have been left behind. This low level DNA is sometimes referred to as touch DNA. It can even be collected from a victim's skin or bruises where they were handled roughly. Low level DNA samples may be helpful when examining evidence where it would be difficult to retrieve fingerprints, such as textured surfaces on gun handles or automobile dashboards. However, not all jurisdictions have the capability to process this evidence. To compare the victim's suspect's DNA profile to the recovered crime scene DNA, the laboratory will need to have their known biological samples available for a side-by-side comparison. These known samples are called reference samples. In some jurisdictions, a DNA sample is routinely taken from an arrestee during the process of booking and fingerprinting. However, there is an evolving area of law, and states vary in their laws governing the collection of DNA from arrestees. Sometimes a court order is required to retrieve a reference from the person of interest. Reference samples are always collected from victim unless they choose not to cooperate with the investigation. In that case, court order might be required. Reference samples are often collected by swabbing the inside of the cheek. Determining a DNA profile, forensic scientists can use DNA profiles to identify criminals or determine parentage. A DNA profile is like a genetic fingerprint. As we know, every person has a unique DNA profile, making it very useful for identifying people involved in a crime. The only exception to this is identical twin. While discussing more DNA profile in solving crimes, now, the result from DNA profile may be used in court. For example, the samples collected from a crime scene might match the DNA of a suspect. This could be used as evidence that the suspect had been present at the crime scene, but it does not necessarily prove that the suspect committed the crime. So, DNA evidence is rarely the sole basis of a prosecution case. It is most useful when placed alongside other evidence such as fingerprints, footprints, crime scene examination, and eyewitness accounts. Other biological evidence may also be collected, for example, blood splash patterns. um, Showing the direction of injury and microbial information which may give clues as to the time of death. How and where DNA testing is performed? A question arises in our mind. DNA testing must be conducted in a laboratory, which dedicated facilities and equipments that meet the FBI's stringent QA's requirement. Most publicly funded DNA crime laboratories in the US are part of state, regional, or municipal law enforcement agencies and accept submissions from multiple agencies. Prior to performing DNA analysis at the laboratory, initial testing is often conducted at the crime scene to determine the type of biological material in question. Screening for the presence of biological material may also be conducted in the laboratory to determine if a specific biological fluid may be present. Most biological screening tests are presumptive in nature and do not specifically identify a bodily fluid. To determine who deposited biological material at a crime scene, unknown samples are collected and then compared to known samples taken directly from a suspect or victim. Most DNA samples submitted to a laboratory undergo the following processes: extraction, that is the process of releasing the DNA from the cell, then actually the quantitation, and then the amplification, and then separation, last but not least analysis and interpretation, and finally quality assurance. And here we will study about quantitation. One of the standards all DNA testing laboratories must meet is to ensure that the DNA recovered from an extraction is human rather than from another source such as bacteria. This is done through quantitation, where the quality and quantity of DNA present in a sample is measured and assessed. Additionally, determining the amount of DNA in a sample is essential for success in the next step, since most amplification systems require a narrow range of input DNA. This sample is completed at BCA through the use of purchased quantifier DNA human quantification kit, and then running all the sample in the instrument known as ABI Prism 7500 sequence. All samples and detection system. This process takes approximately 30 to 60 minutes to set up and then approximately 2 hours to run on the instrument. Next is the amplification. DNA amplification is accomplished through the use of a technique known as polymerase chain reaction (PCR). PCR is a process in which millions of copies of a specific sequence of DNA can be made in a matter of only a few hours. This is important for forensic DNA analysis, since the DNA often found at crime scenes is limited in both quantity and quality. This molecular zeroing process is completed by precise heating and cooling of the sample in a thermal cycling pattern for approximately 28 cycles. Next is the capillary electrophoresis. After the PCR reaction is completed, it results in a large mixture of amplified DNA molecules which need to be separated in order to distinguish the various molecules from one another. This is accomplished through a process known as capillary electrophoresis. DNA molecules carry a negative charge, and once an electric current is applied to the sample, the molecule enter a very thin capillary filled with a gel-like polymer and migrate towards the positive anode at the other end of the capillary. The PCR products are then separated by size because the smaller DNA molecule will have an easier time migrating through the polymer than the larger DNA molecules. The data from this process is then collected on a computer attached to the CE instrument, and then through the use of a software program, a DNA profile is developed. The BCA uses an instrument called ABI Prism 3130 genetic analyzer, which is capable of analyzing 4 samples at a time. Setup for this step takes approximately 10 to 15 minutes, and each injection on the 3130 takes approximately 45 minutes. Next is the separation. Separation is a process separating Separation is actually the process of separating amplified DNA product to permit subsequent identification. Analysis and interpretation is the process of quantitatively and qualitatively comparing DNA evidence samples to known DNA profiles. Quality assurance is a process of reviewing analyst reports for technical accuracy. How the results are interpreted. The DNA analysis process provides the analyst with a chart called an electrophoregram, which displays the genetic material present at each loci tested. Each of the gray bars on the graph that will be shown in the next slide, except for the last one, corresponds to a locus. The final gray area is used to indicate the gender of the individual. In a complete profile, each person will exhibit either one or two peaks, known as the alleles, at each locus. The following electrophoregram is an example of a profile from a single individual, single source profile. This is the full DNA profile, electrophoregram data generated. Loci that displays only one allele indicate that the individual inherited the same markers from both parents. At this locus, where two alleles are displayed, the individual inherited different markers. This electrophoregram displays match between two single source profiles. One is the evidence sample and the other one is suspect standard. The next slide is comparing profiles against a central database. To enable profiles to be searched against a large national database, the FBI created the National DNA Index System in 1998. This national database is part of the combined DNA index system that enables law enforcement agencies throughout the nation to share and compare DNA profiles to help investigate cases. As of 2012, there are more than 10 million DNA profiles in the system, and CODIS has produced leads that have assisted in almost. Once a laboratory enters a case into CODIS, a weekly search is conducted of the DNA profile in NDIS, and resulting matches are automatically returned to the laboratory that originally submitted the DNA profile. CODIS has 3 levels of operation: local DNA index system, state DNA index system, national DNA index system. The national DNA index system databases contain DNA profile from convicted offenders, DNA profile of individual convicted of crimes, arrestees profile of arrested person, if state law permits the collection of arrested sample, forensic announced DNA profiles of unknown individual developed from crime scene evidence such as semen stains or blood. Missing person contain DNA reference profiles from missing persons. Biological relatives of missing person contain DNA profile voluntarily contributed from relatives of missing person. Unidentified human remains contains DNA profile developed from unidentified human remains. So each database has its own rules regarding the number of STR markers that must be present for the profiles to be uploaded. As of January 1, 2017, the National DNA Index System requires that 20 autosomal STR markers be tested, and the profile must contain information for at least 10 loci. The requirements are less stringent for state and local databases. States require the profile to have information for 7 or more loci, and the local databases requires at least 4 loci to be present or to be uploaded. DNA databanks. There is a New Zealand databank of DNA profile. It contains over 70,000 DNA profiles of convicted offenders and some volunteers. This national databank collection can be matched against DNA profiles collected from unsolved crimes. How to test for your father's DNA? See, each one of us inherits randomly 23 chromosomes. from her father and another 23 from her mother. Chromosomes are found in certain areas of our body, including white blood cells, cells inside our cheek, and in saliva and hair roots. DNA collected from any of these sites can be used to identify people and determine relationships. Paternity test DNA examines certain loci on the chromosomes. Comparing the DNA sequence of a child to that of an alleged father can show if the child's DNA was derived from that man or not. When possible, the mother should also be included in the testing since she can provide additional genetic information and simply the testing process. Paternity can still be checked without the mother's DNA. So, the first step with paternity test DNA is to see that if the man can be excluded, there are certain genetic markers that a father must transmit to his children. Similarly, there are certain markers in the child that must come from the father. If any of these fundamental requirements are not met, the man cannot possibly be the father. If a man cannot be excluded, a probability of paternity is calculated and reported. The probability cannot be 100% since every man in the world is not being tested, but a probability value of 99.99% or higher eliminates most other men from being the biological father. This virtue proves that the tested man is indeed the father of the child. The next is importance of DNA in sexual assault cases. So, it's not easy, says David Foran, but it's not impossible. DNA evidence that is recovered uh, from explosive devices is not usually high quality. It mostly comes from fingerprints left by whoever handled the bombs and the containers, like suitcases and backpacks where the devices were placed. Fingerprints melt quite easily with heat because they are made of skin oils, but they often contain skin cells the body sheds throughout the day. Even if the fingerprint melts, skin cells are more durable. Those skin cells are used by investigators to recover DNA evidence, but it's not exactly high quality. The cells are dead, so the DNA is likely to be degraded. To make things worse, it's likely that objects recovered from a messy blast scene have a mixture of multiple people's DNA, victims as well as perpetrators. The challenge is then to separate the DNAs to identify the suspect or suspects. It's like pouring four different colors of paint into a jar and stirring them up. You cannot differentiate them. The odds of success could increase if investigators already have DNA records of potential suspects and can use those to differentiate the multiple DNA traces. The question arises that what is the future of forensic DNA analysis? The field of forensic DNA testing is headed for the next decade are provided in the context of where the field has come over the past 30 years. Similar to the Olympic motto of faster, higher, stronger, forensic DNA protocols can be expected to become more rapid and sensitive and provide stronger investigative potential. New short tandem repeat STR loci have expanded the core set of genetic markers used for human identification in Europe and the USA. Rapid DNA testing is on the verge of enabling new applications. Next generation sequencing has the potential to provide greater depth of coverage for information on STR alleles. Familial DNA searching has expanded capabilities of DNA databases in parts of the world where it is allowed. Challenges and opportunities that will impact the future of forensic DNA are explored, including the DNA for education and training to improve interpretation of complex DNA profiles. Thank you so much.